Today we'll see beta oxidation of fatty acids and ketogenesis. First we'll see what is beta oxidation and what are the stages of beta oxidation, and finally then we'll know the energetics of beta oxidation. Many times the question, explain beta oxidation of palmitic acid and add a note on its energetics, has been asked in the university examination. So what is beta oxidation? Beta oxidation may be defined as the oxidation of fatty acids on the beta carbon. And this results in the sequential removal of two carbon fragment acetyl-CoA. Fatty acid oxidation stages. The beta oxidation of fatty acids involves three stages. First one, activation of fatty acids occur in the cytosol. Second one, transport of fatty acids into the mitochondria. Third one, beta oxidation proper in the mitochondrial matrix. Fatty acids are oxidized by most of the tissues in the body. However, brain, erythrocytes and adrenal medulla cannot utilize fatty acids for energy requirement. First step, the activation of fatty acids. The fatty acids are activated to acetyl-CoA by the enzyme thiokinases. The reaction occurs in two steps and requires ATP, coenzyme A and magnesium. Fatty acid reacts with ATP to form acetyladenylate, which then combines with the coenzyme A to produce acetyl-CoA. In the activation, two high-energy phosphates are utilized since ATP is converted to pyrophosphate. Then enzyme inorganic pyrophosphatase hydrolyzes inorganic phosphate to phosphate. The immediate elimination of inorganic phosphate makes this reaction totally irreversible. The second step, the inner mitochondrial membrane is impermeable to fatty acids. A specialized carnitine carrier system, also known as carnitine shuttle, operates to transport activated fatty acids from cytosol to mitochondria. And this occurs in four steps. The first one, acyl group of acetyl-CoA is transferred to carnitine, catalyzed by the enzyme carnitine acyl transferase 1 that is present on the outer surface of inner mitochondrial membrane. Second step, the acyl carnitine is transported across the membrane to mitochondrial matrix by a specific carnitine or carrier protein. Third step, carnitine acyl transferase 2 enzyme that is found on the inner surface of inner mitochondrial membrane converts acyl carnitine to acetyl-CoA. And fourth step, the carnitine is released and returns to cytosol for its reuse. Third step, beta oxidation proper. Each cycle of beta oxidation liberating a 2-carbon unit acetyl-CoA occurs in a sequence of 4 reactions. First one, oxidation. Second one, hydration. Third one, oxidation. And fourth one, cleavage. Oxidation. Acetyl-CoA undergoes dehydrogenation by an FAD-dependent flavor protein or flavor enzyme, acyl-CoA dehydrogenase. A double bond is formed between alpha and beta carbons, that is the second and third carbon atom. Second, hydration. Enol-CoA hydratase, the enzyme, brings about hydration of double bonds to form beta-hydroxyacyl-CoA. Third one, oxidation reaction. Beta-hydroxyacyl-CoA dehydrogenase catalyzes the second oxidation of and generates NADH. The product formed is beta-ketoacyl-CoA. The fourth reaction, cleavage. The final reaction in beta oxidation is the liberation of a 2-carbon fragment, acetyl-CoA from acyl-CoA. This occurs by a thiolytic cleavage catalyzed by the enzyme beta-ketoacyl-CoA thiolase. The new acyl-CoA formed, containing 2 carbon atoms less than the original, re-enters the beta oxidation cycle. The process continues till the fatty acid is completely oxidized. Now the energetics. 7 cycles of beta oxidation will occur, resulting in 7 FADH2 and 7 NADH. And as a result of this, 14 and 21 ATPs will be formed through electron transport chain and oxidative phosphorylation. At the end of 7 cycles of beta oxidation, 8 acetyl-CoA will be synthesized. And through this 8 acetyl-CoA, 96 moles of ATP will be yielded. A total energy from 1 mole of palmityl-CoA will yield 131 ATP molecules. Out of this, 2 molecules will be utilized for activation, that is the formation of palmityl-CoA. A net yield for 1 molecule of palmitate will be 129 ATP molecules. Next one, ketogenesis. The compounds, namely acetone, acetoacetate, and beta-hydroxybutyrate, are known as ketone bodies. And these ketone bodies are water-soluble and energy-yielding. Synthesis of ketone bodies, or ketogenesis. The synthesis of ketone bodies occurs in the liver. The enzymes for ketone bodies synthesized are located in mitochondrial matrix. Acetyl-CoA formed by the oxidation of fatty acids, pyruvate or some amino acids is the precursor for the ketone body synthesis. Ketogenesis occurs through the following reactions. First one, 2 moles of acetyl-CoA condense to form acetoacetyl-CoA. This reaction is catalyzed by thiolase, an enzyme involved in the final step of beta oxidation. Hence, acetoacetate synthesis is appropriately regarded as the reversal of thiolase reaction of fatty acid oxidation. Second one, acetoacetyl-CoA combines with another molecule of acetyl-CoA to produce beta-hydroxy-beta-methyl glutyl-CoA. In short, it is HMG-CoA. HMG-CoA synthase, the enzyme catalyzing this reaction, regulates the synthesis of ketone bodies. HMG-CoA lyase cleaves HMG-CoA to produce acetoacetate and acetyl-CoA. Acetoacetate can be undergone spontaneous decarboxylation to form acetone. Fifth step, acetoacetate can be reduced by dehydrogenase to beta-hydroxybutyrate. Next is utilization of ketone bodies. The production of ketone bodies and their utilization becomes more significant when glucose is in short supply in the tissues, as observed in starvation and diabetes mellitus. During prolonged starvation, ketone bodies are the major fuel source for the brain and other parts of the central nervous system. The reactions of ketone bodies: beta-hydroxybutyrate is first converted to acetoacetate and metabolized. Acetoacetate is activated to acetyl-CoA by a mitochondrial enzyme thiophrase. The coenzyme A is donated by succinyl-CoA, an intermediate in the TC cycle or citric acid cycle. Thiophrase is absent in liver, hence ketone bodies are not utilized by the liver. Thiolase cleaves acetoacetyl-CoA to 2 moles of acetyl-CoA. The acetyl-CoA can be utilized for the energy purposes. Thank you.